And we're here now with Jeff Chao. He's over at the FMI in Switzerland. Thank you so much for joining us, Jeff.
No, thanks for having me.
Your work involves imaging mRNA molecules in living cells. What insights have you gained about how these cells are regulated?
Yeah, so it's been really exciting. I think for the first time, we've been able to go beyond just being able to see the molecules inside cells. I mean, that's also just exciting by itself. You can watch individual molecules of RNA moving around inside cells. But now we've developed tools that you can really ask questions about what's going on in their lives. So not just, looking at them, be able to see what they're doing and figuring out how they're being regulated. And so two key aspects in an RNA's life are, when they translate, when they make proteins, and also when they decide that it's time to stop making proteins and degrade. And so my group's really been interested in trying to develop tools that allow us to pinpoint these moments in their lives and be able to ask, how much, protein do they make? When do they decide, okay, now it's time to degrade, we don't need the RNA anymore? And so really, there's been lots of surprises here. When we were looking at stress, so when cells get stressed, they form these compartments inside the cell called stress granules. And for a long time, people thought that RNAs that were inside these compartments were completely translationally repressed. And because we really only had measures that allow us to look at the ensemble of RNAs inside the cell, and usually you had to lyse cells and then infer what they were actually doing inside the cell. And so with the tools we have now, we could go in there and look at the individual molecules of RNA, and what we could find is that actually these stress granules, despite the fact that the dogma in the field is that this was a compartment that was translationally repressed, we can see that the RNA is translated there just fine. And so I think that just sort of highlights the questions that you can address now with these sort of tools. And so I think it's an exciting time.
What techniques do you use to study this?
Yeah, so we actually use fluorescent microscopy, and so we develop tags that we can put on these individual molecules and follow them around. And then, like I said, we try to do more than just observe them, and so we develop tools that allow us to look when they degrade. So we set up a system that the RNA is labeled in two colors, and essentially when one of the colors disappears, we know when the RNA gets degraded. And so we've been playing games like this to understand how RNAs are regulated.
Interesting. We have a video from you demonstrating this. Can you explain what's happening here?
Sure. So in this video, what you see is a dual-colored RNA molecule. You'll see it in magenta and green, and you see it sort of diffusing around. And then there's a moment when the RNA gets cut in half, and then you can actually track the two halves of the molecule in real time. And this was surprising because oftentimes people thought that the RNAs might be degraded on the ER, but we could actually see this is going on everywhere inside the cell. And so these tools allow us to sort of address some of these questions that we could really only imagine how they actually worked. And so now I think it's exciting because we can try to image these processes in live cells.
Sounds very informative. Thank you so much, Jeff Chao.